very noisy due to the background noise being amplified. Second, data could potentially be missing. This would result in uneven transcript coverage and gene dropouts. Typical analysis methods for single-cell RNA-seq data involve quantifying gene expression for each cell, followed by clustering gene expression data to identify specific cell types. However, there is very limited focus on reconstructing transcript sequences. Existing assembly methods are only limited to reconstructing specific gene targets, such as B-cell receptors and T-cell receptors. Although de novo bulk RNA-seq assemblers such as Transcript and Trinity may be used, but these tools are not designed to address the challenges in analyzing single-cell RNA-seq data. So here it's a hypothetical example showing the effect of amplified noise and uneven transcript coverage. And as you can see, using a bulk RNA-seq assembler, you will almost always result in fragmented assembly. And no form of optimization of the assembly parameters would be able to resolve this issue. However, if we pull the reads across multiple cells, it is possible to reconstruct the entire transcript from beginning to the end. But the problem with this approach is you would lose the cell's specificity of the transcript. So a solution to this problem is to borrow reads from other cells only during the process of joining read pairs together for each cell. So this example only consists of 3 cells. So imagine that you're trying to analyze a lot more cells. So that means we need a method that could store the k-mers from many cells in the memory efficiently. And to address this problem, I have developed a program called RNA Bloom, which is a de novo RNA-seq assembler that uses a very compact data structure called Bloom filters for the storage of k-mers and k-mer counts in memory. This tool is designed for assembling bulk RNA-seq data and single-cell RNA-seq data. It is very fast and it is very memory efficient. So a Bloom filter is basically just a binary array, so an array of ones and zeros. It is used for set membership tests. So 1 bits in the binary array are used to indicate items that may be in the set, and at least 1 0 bits are used to indicate items that are not in the set. So here it's a very simple example. So let's say we have a set of 2 items, so E1 and E2, and we assign 3 hash values for each of these elements in the set. So these hash values are used to specify the positions in the binary array where the positions are set to 1. But however, due to hash collision, false positives may exist in Bloom filters. So a Bloom filter de Bruijn graph could be used to store all the k-mers in the data set. And in order to traverse through the de Bruijn graph, it's just a matter of asking the Bloom filter for the existence for all 8 possible adjacent k-mers. And because we are dealing with transcriptome data, storing the k-mer counts is really important for RNA-seq assembly, because the k-mer counts could be used to guide the transcript reconstruction process, and it could also be used to identify noise and sequencing artifacts. And because of that, we also need a method to store the k-mer counts efficiently in memory. So to do that, we use a data structure called k-mer counting Bloom filter. So basically the way it works is it stores the k-mer counts as 8-bit miniflow in a byte array as opposed to an array of ones and zeros of a regular Bloom filter. So what's special about this 8-bit miniflow data type is that it could represent exact counts between 0 to 16. And for numbers larger than 17, it will use approximate counts. So as shown by the figure on the right, you can see very clearly that the approximate counts do in fact represent the true counts really well. And you can also see that the approximate counts in different orders of magnitude are easily distinguishable from each other. And this is very important in transcriptome assembly. So now that I have told you about the data structure that is used in RNA Bloom, so I'll go over how RNA Bloom assemble transcripts. So its workflow is basically consists of 3 stages. First, it will construct the Dubun graph, followed by reconstruction of read fragments, and finally, it will reconstruct transcript sequences from the reconstructed fragments. In the first stage, to construct the Dubun graph, basically you just take all the reads in your data set, you break it into k-mers, so which is a subsequences of length k, and these k-mers are used to populate the Dubun graph Bloom filter and the k-mer counting Bloom filter, which I have just told you about. And at the same time, k-mer pairs along the reads are also stored in another Bloom filter. So after these 3 Bloom filters have been constructed and stored in memory, we can reconstruct read fragments from pair-end reads. So we can use the 3 Bloom filters to correct errors in the reads. And after that, we can use the Bloom filters to find the path that is most supported by k-mer pairs from the reads to join the 2 reads of a pair read together. And after the fragment has been reconstructed, we can store another set of k-mer pairs in another Bloom filter. So it's important to note here that these k-mer pairs are at a much longer distance than the ones from the reads. So after all the fragments have been reconstructed for the entire data set, the fragments could be assigned to one of 14 strata based on the fragment's minimum k-mer count and its length. And this is done to account for the different magnitude of expression. And it's also used to identify fragments that may be too short for extension in the next stage of the RNA Bloom. So finally, in the last stage of the program, it will attempt to extend the reconstructed fragments in both directions. So basically, the way it works is it will use the k-mer pairs from the reads and the k-mer pairs from the reconstructed fragments to guide the reconstruction process. And this is how transcripts are reconstructed in RNA Bloom. And in the context of reconstructing transcripts for single-cell RNA-seq data, as mentioned before, I propose a solution to pool the reads together from multiple cells. And in order to do that in RNA Bloom, I construct a shared Dubun graph using the reads from multiple cells. Once the graph has been constructed, we can use that to reconstruct fragments for individual cells. And after the fragments have been reconstructed for individual cells, the shared Dubun graph is discarded from memory, and a new Dubun graph is generated for individual cells. And those Dubun graphs would be used to reconstruct transcript sequences for individual cells, and this is how the cell's specificity of transcript is maintained in RNA Bloom. So I perform benchmarking of RNA Bloom using a 7-cell dataset, which consists of 7 mouse B1A cells. And I compare my program RNA Bloom against 3 bulk RNA-seq assemblers, Transibis, Trinity, and RNA Space, which is an assembler based on the Space algorithm for single-cell genome assembly. And for these 3 assemblers, they reconstructed transcript sequences for each cell individually. And to evaluate the assembly, I use a program called RNA Cross, and I also use error-corrected nanopore reads. So this table here shows that using 12 CPUs, my program RNA Bloom uses the least amount of memory, and it is the fastest among the 4 approaches evaluated. And this figure shows that RNA Bloom has the highest sensitivity across all 7 cells. And it's important to note that RNA Bloom assemble 2.6% to 8.4% more known isoform than the next best approach. And this figure shows the number of misassemblies for all 4 approaches. And we can see very clearly that RNA Bloom made the least misassemblies for 6 out of 7 cells. And for the publication where this data set was released, the authors of this publication identified that Trinity, a bulk RNA-seq assembler, would generate a lot of isoforms that are not detected by ONT reads, and its assembly is quite fragmented in some cases. And now if we look at the assembly of RNA bloom for the same gene, for the same data set, we can see that RNA bloom is able to reconstruct the different isoforms for this gene to near full length. And it's also important to note that RNA bloom is able to reconstruct a novel isoform that is actually supported by nanopore reads. So now I want to show some preliminary results where I apply RNA bloom to a much larger data set. So this data set consists of 260 cells. So these cells are mouse embryonic stem cells, and they were derived from 3 Fluidigm C1 chips. And as you can see from this table, this data set has a lot of reads. So on average, each chip would have about 1.2 to 1.3 billion reads, and in total there are about 3.9 billion reads. And I use RNA bloom to assemble the transcriptome data for each chip separately. So the reason why I picked this data set is because the authors of the publication where they released this data set, they had identified 3 subpopulations based on gene expression analysis of 86 pluripotency factors and differentiation markers. And based on their analysis, they determined that these 3 classes of cells correspond to different pluripotency states of the mouse embryonic stem cell. So I wanted to evaluate the same data set from a sequence assembly perspective. So here is a table that shows the performance of RNA bloom for the assembly of the 3 chips that I mentioned earlier. And we can see that RNA bloom is able to reconstruct the transcript sequences for a chip using about 30-something to 40 gigabytes of memory. And using 12 CPUs, it will take a little bit less than 3 days. So as mentioned before, I wanted to evaluate this data set from a sequence assembly perspective. So I wanted to perform clustering based on isoform reconstruction, and wanted to compare that to the gene expression results that were presented by the authors of this data set. And to do that, I measure the normalized isoform reconstruction. So what that means is I will normalize the length of the isoform to between 0 and 1. And to calculate gene reconstruction, I would take the maximum isoform reconstruction for the corresponding gene. And after that, I will filter out any isoform that has very low variance in reconstruction across all the cells in the data set. And finally, I will perform hierarchical clustering. So this is a heat map showing the gene reconstruction for all the genes that were evaluated by the authors. And this is without clustering. And we can see that by looking at just gene reconstruction alone, we can see that the pattern seems to agree with the ones derived from gene expression analysis. And now if we perform clustering for both the genes and the cells, we can see that the clustering is somewhat different from the gene expression analysis. But this sort of makes sense because I'm only looking at the data from a sequence-based perspective. I'm not looking at the gene level expression or even isoform expression. And it's also important to note that so the cells that are colored in yellow here has the ability to go back into the pluripotent state, which is indicated by the green color here. So it's possible that for some of these cells are being transitioned from one cell type to the other. And here it's a heat map showing the clustering based on isoform reconstruction. And we can see that the clustering show very similar results to gene reconstruction. So in conclusion, I have shown that by pulling reads from multiple cells, it is possible to improve the transcript reconstruction for individual cells. And I have shown that RNA bloom is very fast and it doesn't use a lot of memory. This is because it uses a data structure called bloom filters for storage of k-mers in memory. So I would like to thank my colleagues, Riedman, Justin, Renee, and my supervisor, Dr. Inashborough, who has been guiding me through this research project. And I also want to thank Ben, who is in the audience, and Lauren, who is my travel buddy for the trip for ISMB, and I thank her for being supportive this whole time. And I want to thank the author of the mouse embryonic stem cell dataset, and I thank her for helping me understand this dataset. And I want to thank my funding sources, ISMB, and I thank ISCB for awarding me the Travel Fellowship Award. And once again, my name is Ming Nip. You're welcome to contact me through email. And this is this tool, RNA Blooms, is actually released publicly on GitHub, and you could find it on that link right there. You could just go into the release section of the GitHub page, and you could download the tool. So there is nothing to install. You could download the tool, and there are instructions on how to run it. And I would also like to draw your attention to my colleague's poster, which is happening in the afternoon. And in fact, my colleague Ben will be giving a talk right after lunch. So please do come back after lunch and show up on time. Yeah, that's all I have for you guys today, and I thank you for your attention.